Hello, and welcome to DNA, RNA, and everything in between, featuring Danny, Lily, Sammy, and Eva. Today we'll be talking about DNA replication and protein synthesis, so sit back and relax. First off, we will begin with DNA replication. The reason DNA replicates is because of the 3 R's and a G: regenerate, repair, reproduction, and growth. Before we talk about replication, let's tell you what DNA actually is. DNA, or deoxyribose nucleic acid, is a double-stranded molecule that contains a nucleotide that is composed of 3 parts: a phosphate group, a pentose sugar, and a nitrogenous base. Those bases are adenine, thymine, guanine, and cytosine. DNA only found in the nucleus. DNA replication has many key components. These components are enzymes. The first enzyme we'll be talking about is helicase. The job of helicase is to break the weak hydrogen bonds between the paired bases to unzip the double helix. Next is primase. Primase sets the primers and or start points by snatching the nucleic acid so the polymerase knows when to start building.
The polymerase will then start rebuilding a new strand by pulling nucleotides within the nucleus that fit together. Through the process of complementary base pairing, it also attaches nitrogenous bases to the sugar backbone. Polymerase can only work in one direction, creating a leading and a lagging strand. Lagging strand must jump ahead and work backwards, leaving gaps. These gaps are called Okazaki fragments. Ligose will then connect the fragments and seal any breaks in the sugar phosphate and backbone. Unlike DNA, RNA, ribonucleic acid is found in the nucleus and cytoplasm, and containing all the nitrogenous bases, but instead of thymine, it contains uracil.
Protein synthesis is the transformation of DNA to RNA, then to a protein by the process of transcription and translation. Transcription occurs in the nucleus and turns DNA to mRNA, then it exits through a nuclear pore. The first step of transcription is called initiation. This stage begins with the RNA polymerase separating the strands of DNA at the specific region called the promoter. It bonds the RNA nucleotides through complementary base pairing along the DNA template, as only one strand of DNA is needed for RNA synthesis. This RNA polymerase is similar to the DNA polymerase during the DNA replication, as it only forms polynucleotides in the 5-3 direction. RNA polymerase differs from the DNA counterpart as it doesn't need a primer to start the chain. The second step of transcription is called elongation. An RNA polymerase travels along the DNA template strand after the unwinding of the DNA, and it begins to synthesize the single strand of messenger RNA, also known as mRNA, by adding the complementary bases. These complementary bases are different in RNA, as instead of thymine, uracil pairs with adenine instead. After the point of RNA synthesis, the double helix is reformed and the mRNA molecules are stripped away, causing an elongation of the mRNA as this process continues. The third stage is called termination. The transcription process will continue unless the RNA reaches its terminator sequence. The newly formed mRNA sequence detaches itself from the DNA out of the nuclear pore and into the cytoplasm. It is then translated from its mRNA state into an amino acid sequence. The length of the transcription is by its start and stop sequence and equals approximately 1 gene. In summary, transcription is when DNA splits into mRNA, then is sent outside of the nucleus through a nuclear pore where it is then translated into an amino acid sequence.
The secondary process of protein synthesis is called translation, which is a process of the mRNA to a protein. This stage of protein synthesis occurs with ribosomes, organelles that are non-membrane bound, small in size, and composed of protein and RNA. These ribosomes are found in the cytoplasm on the surface of the endoplasmic reticulum. They read from a triplet code known as a codon that are then interpreted by the cell. Many of these codons together begin reading by the cell, which forms a polypeptide. The mRNA is taken from its nucleic acid form to its amino acid translated form. In order for mRNA to reach this stage, it must be translated by its translator, tRNA. tRNA brings the amino acids to the ribosome, where it is brought to the expanding polypeptide chain. The tRNA holds 3 nucleotides called anticodons, which bind to the nucleotide of a codon through base pairing. The first step of translation is a process called initiation. During initiation, the mRNA binds to the ribosome as the ribosome checks it for the start codon, a 3-letter code, AUG, signaling the beginning of a translation. After that, a tRNA will align the complementary anticodons while the codon of the mRNA fills with the amino acid. Another tRNA will go along the second codon. During this, the ribosome checks the anticodons and codons for matches, then causes a peptide bond with the two tRNA amino acids. The ribosome continues to travel along the mRNA the space of one codon. The next stage is elongation stage. And during this stage, the ribosome follows along the mRNA, identifying the codons, then binding amino acids with tRNA. There are enzymes along the ribosome, further the amino acids, and then large chain expands. The final stage of translation is the process of termination. Termination occurs when ribosomes find a stop codon. A three-letter code includes UAG, which causes the translation process to stop or terminate. During this process, there is a release of the completed polypeptide chain and a release of the protein, which then forms into secondary or hereditary shapes. The ribosome then disassemble, which result in mRNA disintegrating.